Thank you very much, Donata. Thank you, Bonka, for helping me out. Thank you for your invitation to this EPTRI platform and also for this meeting. As Donata said, I would like to present this Placenta Platform, which is actually a thematic research platform dedicated to placenta studies under EPTRI aims. And the aim of this platform is to fill the gap and provide know-how and technologies and access to in vitro and many models to study placental physiology, pathology, pharmacology, and toxicology. So during my presentation, I would like to introduce the current teams that will work together on this platform. I would then try to stress out the importance of the placenta for fetal development and programming, and also the methods to study the placenta that we have currently available in our teams. So I'll start off with introducing my colleagues and friends, actually placentologists, that are now enlisted on this Placenta Platform. Professor Christiane Albrecht is from Bern University in Switzerland. Professor Kathy Valancourt is from Montreal in Canada. Professor Udo Market is from Jena Klinikum University, Germany, and myself, Charles University, Czech Republic. All these teams have got decades-long experience in placental research, and we have already established a close collaboration among these teams. So I'm hoping this is just the beginning, and hopefully if we get funding, we can grow, because there are many more great teams within Europe and abroad focusing on placenta research. Why to study placenta? Why do we need to know something about placenta? Because I mean, this EPTRI platform, obviously it's a pediatric platform focused on treating newborns and babies after delivery, but we shouldn't forget that what happens before delivery is very crucial for later life outcomes, and the prenatal period is the foundation for later life outcomes, and placenta functions are crucial to maintain optimal in utero conditions for proper development during pregnancy and also for programming. I'll mention a little bit later. Of course, if there is some placenta malfunction, it will result in not optimal in utero conditions that we can see maybe straight after delivery, such as malformations, fetal growth restrictions, or sometimes we don't see anything in the delivery room, but still the placenta malfunction is somehow reflected into chronic effects, such as diseases developed later in life, metabolic diseases, psychological, mental diseases. It's actually discipline called Developmental Origins of Health and Disease, DOHAD in short. Many researchers prefer to study brain and kidney and the lung, liver, and many other fancy organs to investigate, because they think they're like the most important in their research and in life. But we should really not forget that the placenta is the first fetal organ to be created, and then comes the rest. If there is no placenta, there is no pregnancy, there is no baby, there is no delivery. Placenta is really crucial from the very beginning. It's the first fetal organ, and during this 9 months of pregnancy, it fulfills many vital roles for the fetus, for its development, programming. So it provides it with nutrition, immunoprotection, oxygenation, endocrine functions, and we don't know all this picture yet. We still have to study it and see how it all works. When I talk about placenta to general public, but also to researchers, I try to compare it to a symphonic orchestra and use this musical analogy, because you are looking now at an orchestra of several individual musicians with different instruments, and they all play, they have to stay in harmony and key and tune and tempo. And then if everything works fine, then the audience can enjoy the music, the harmony of the music. But if something goes wrong with the conductor, he gets sick, drunk, intoxicated, tired, whatever, then even the musician may get lost or crazy, and the orchestra gets out of tune, out of tempo, out of harmony, and then the audience is disappointed because we don't see or we don't hear beautiful music. And this is actually how it works also in the pregnancy. Placenta, as a conductor, creates many links with individual players in the ensemble. And if you read the recent literature, you can find terms such as placenta brain axis, placenta heart axis, placenta lung axis, because placenta communicates with all organs and tries to take care of them for successful development. Developmental origins in health and disease have been actually properly described. And in brief, what they say is that if there is some trouble in pregnancy, such as diseases, pathologies, pharmacotherapy, these can be epidemiologically clearly linked to diseases later in life. This is what we call fetal programming, or DOHAT, Developmental Origins of Health and Disease. So these epidemiological links have been clearly established starting in 1990s, continuing nowadays, but the mechanistics in between, what is the real cause of these troubles, has been not properly described. Of course, we could blame placenta for not providing the proper or optimum in utero conditions for fetal development and programming. So maybe placenta is the organ to blame, or at least partly. So we have to study the placenta. We need to look how it works, and how it works on the normal condition, and how it works under disrupted conditions. So we need to study placental physiology, pathology, but the problem is that it's really very difficult ethically and technically. We cannot, of course, we cannot investigate placental function during pregnancy in human. We cannot interfere. We cannot take samples. So we are kind of limited, and we have to perform many other methods and use several methods, alternative methods, to gain some knowledge and then treat the results in a complex manner, so that we can somehow create a picture and draw conclusions from what's going on. And the unique feature of our Placenta platform is the capacity to offer integrated services by the platform partners in a hierarchical cascade, starting with low-cost, high-throughput analysis, followed by ex vivo tissue explants and animal experiments and human ex vivo placenta perfusions, to really be able to achieve some new knowledge. So now I will guide you kind of quickly through the methods that we have established so far. And of course, feel free to ask or contact me by email, whatever, if you need more details, because now we don't have time to go through each and every method. I'll just take it like logically from cell-based in vitro methods. We have various models at this moment available for our research, such as genetically modified MDCK cells, cells derived from the placenta, such as BIVO, JAG, JAR. We have also learned the methods of isolating primary trophoblast cells from the human placenta, and all these techniques can be used for many goals and projects, such as study nutrient transport across the placenta. If we culture the cells in a monolayer, and then we add the substrate, and we see how the substrate is transported, and what is the effect of some inhibitor, for example, some endocrine disruptor, some pharmacotherapy during pregnancy. So we can see some basic information on the in vitro level. Then we also provide a method of dually perfused rat placenta, which at this moment there are only 2 teams in the world. One is in Japan, one is here in the Czech Republic. to study nutrient transport again and homeostasis, for example, of endogenous hormones, glucocorticoids, monoamines. You might wonder how this compares to human placenta, but rest assured, in several projects, it's a really nice correlation between human and rat placenta. In some other projects, we have to be careful, of course, about, for example, expression of some enzymes or transporters, but it's a very, very good and useful model to investigate. For example, as you can see here, placental homeostasis of glucocorticoids, monoamines, and so on. And then, of course, we need to work also with human placenta. As I said, we have limited access to human placenta because we can only take it after delivery, which is when the placenta is completely mature and complete. We have limited access to first trimester placentas, second trimester placentas. But this is what we have to kind of acknowledge and be aware of. But still, we can use this delivered placenta for methods such as villus fragments, placenta explants, placenta homogenates for placental metabolism of drugs or endogenous compounds. So we have all these models in our labs to study placenta physiology, toxicology, pathology. We have also very sophisticated models such as isolation of membrane vesicles. We can isolate membrane vesicles from maternal side of the placenta and from fetal side of the placenta and investigate what's going on the maternal side and on the fetal side. How does it communicate? So this is again available in our labs, and also in our teams, we have this model of perfused human-term placenta lobule system, which is very kind of technically demanding, laborious. So it's kind of a high-end model that we only use if justified and if justified by the previous, for example, in vitro models. And again, we can use it for many projects to study placental behavior, physiology, pathophysiology, and so on. Then definitely we can look also or use animals for chronic exposure to some pathological conditions or pharmacotherapy. So, for example, we can treat animals with drugs that have been used in pregnancy, such as, I don't know, antidepressants or antidiabetics, or we can also subject the animals to pathological conditions, stress, inflammation, and see on the long run what's going on. We can also do pharmacokinetic studies, or currently we have acquired a beautiful Doppler ultrasound machine to see and investigate actually the effect of long-term pathologies or treatment of animals on the placental function. I'm not sure if you can see and hear what we can investigate in our small animals on a tiny level with this high resolution Weber 3100 machine. So I believe this is like a quick look at the methods that we have available. And the reason is, again, I would like to stress out that none of this method can answer all our questions, because placenta is really a complex organ. I know that many people look at the placenta as a garbage after delivery. Baby is born beautiful, but then the placenta comes and we have to throw it to trash and forget about it. That's how we often see placenta, but we researchers, we are confident that placenta is a really very complex organ, and we don't have a single method that would describe all its functions. So we need to use all this or as many methods as possible to draw some conclusion and to understand how placenta works. And if we understand it, we can maybe even prevent pediatric diseases or reduce pediatric diseases taking measures during pregnancy. What our team or teams, these 4 teams, what we are currently working on, because despite this EPTRI platform hasn't been funded, we were disappointed about it, but we still continue working, the 4 teams of us, and now we are focusing mainly on endocrine function of the placenta. We are studying the effect of endocrine disruptors like chemicals, environmental toxins on placental homeostasis, for example, of serotonin and other monoamines, because these monoamines are crucial for fetal development and especially for brain programming. We also look at effect of maternal inflammation on tryptophan catabolism in the fetal placental unit. We have a very well elaborated collaboration with Department of Obstetrics and Gynecology in our university hospital. We also look at the synthesis, degradation, and homeostasis of steroid hormones in the placenta, because believe it or not, placenta also produces and degrades steroid hormones to feed the fetus during pregnancy. And we have over this year we managed to publish several papers. I'm showing you the 3 probably most important that are international papers, including the teams of this Placenta Platform. Basically about what I've said about endocrine function of the placenta. So these are 2020 papers. And lastly, I would also like to show that we managed to get funded by Swiss 3R Competence Center to, as you can see here in the blue screen, we got funded with Christiane Albrecht from Bern to create another model, yet another model of actually placenta, kind of placenta 3D structure to investigate drug transfer during pregnancy. So I also consider it as a nice achievement of our Placenta Platform. And I believe this is all from my side. So I would like to, on behalf of the whole team, I would like to thank you for your attention.
Thanks a lot, Frantisek. I'm sure that everybody appreciated a lot and also enjoyed your very nice presentation. We will share all the presentations on the EPTRI website. And I would also, according to what you said, suggest that maybe even more research units can join this new platform to be settled, and also that further requests for funding can be submitted very soon with the Horizon Europe, where this topic will be for sure included, but maybe also for some networking activities and so on. So thanks again. We will come back to you for further plans to develop the platform.